Hello. Translation is controlled or mediated largely by RNA molecules. The information comes in the form of RNA, that is the messenger RNA. And the translation itself is carried out by rRNA, ribosomal RNA, along with the ribosomal proteins, of course. And there is one another molecule that is central to translation, or it is the molecule that we call it as tRNA, transfer RNA, or it's kind of adapter molecule that is important for reading the message or the information that is present on the mRNA in the form of nucleic acids or nucleotides, and then being able to bring in a corresponding amino acid to the translation so that it can eventually make a sequence of amino acids. So the tRNA molecules play a very central role, and we'll try to understand the importance of it. So in the translation, the tRNAs are called as the adapters because of the obvious reasons, because they are the two languages, language of nucleotide and language of amino acid, and the central molecule that connects between these two, the connection is tRNA. There are usually about 75 to 95 nucleotides in length. They could be many types, and each tRNA can be added by only specific type of amino acid. Of course, one tRNA could recognize multiple codons, but one tRNA will be tagged or added or charged with only one type of amino acid. And there are some of the special things why when the transcription of the tRNA happens, it is produced as a pre-transcript or RNA molecule, and then it is processed. When it is processed, one of the unique features or one of the features is that there are lot of complementarity within the tRNA itself. That means there are a lot of places where they can form hydrogen bonds like this, intramolecular hydrogen bonds. As a result of which, when they base pair like this, they form hairpins and a complex structure such as is shown here. So once it is produced, there are several aspects that happen. The first one is that there is a cut made on the 5' end, and that is done by RNase P. And there is also processing of the 3'. The 3' OH, there is a little bit of cut, and 3 bases are added. That is read from here to this way, that is CCA, because this would have been the 5' side, and this is the 3' side. So we always read the sequence from 5' to 3'. So all tRNAs are added with CCA, and the 3' OH here is the place where amino acid is going to be added onto the tRNA. And there is one more event that happens is the bases that are present in the tRNA are modified. The normal bases are GAUC, like DNA, except the thymine and uracil difference. But once the transcription is done, many of the bases of these tRNAs are modified. So they are modified by addition of methyl groups or rearrangement, something like methyl guanine. D here stands for dihydrouridine, and there are psi stands for pseudouridine. Here T is a methylated uracil, which is normally which is not present in RNA, and it's a nucleotide of the nitrogenous base of the DNA. And there is also one another aspect that happens is some part of the, say something like this whole part of the tRNA is cut out, and that is the event of splicing happens, and now we have a completely mature RNA, tRNA. And For sake of understanding and also to depict their functional importance, these loops, at least in the two-dimensional structure, we can, for the sake of understanding, we can call this as the D loop, because you have so many dihydrouridines. And before that, we will just have a look at what is dihydrouridine and pseudouridine out of many other nitrogenous base modifications. We'll just look at these. So here is uracil. If we look at it, and it is normally we would have had the glycosidic bond here. The ribose that is present would have been present here. This is to the first, the nitrogen at the first position of uracil. But we have the ribose, the glycosidic bond forming to the fifth carbon, which is not, although the chemical formula would remain the same, the organization is different. That's why we refer to it as pseudouridine. And similarly, we also have another one is the two extra hydrogens that are present here. Normal uracil wouldn't have these. So we are adding two hydrogen atoms, so we call it as dihydrouridine. So these are simple modifications that are present on the uracil. Similarly, methyl guanine is also there, and many other aspects, something like that. So because we have a large number of dihydrouridine, we call it as the D loop, and this is the location where the anticodon loop is present. This is where the anticodon is present, which is going to base pair with the codon. So we call this as the anticodon loop. And here we have a small variable loop. We call it as variable because in different tRNAs it varies in size and etc. Some of them I think don't have it as well. And here we call it this loop as pseudouridine due to the presence of pseudouridine. And the another important one is the acceptor arm. All this arm is called as the acceptor arm. The CCA is added by an enzyme, and this is where the amino acid is going to be attached. So because it is going to accept the amino acid, we call it as the acceptor arm. So these structures are important for multiple purposes, although in translation we would just discuss mostly about the anticodon loop and the acceptor arm, because they are the ones that are recognizing the codon and also bringing the amino acid. But other structures also have several important functions to be recognized as tRNA by its different enzymes, and also when they fit into the ribosome too, they have important roles. I hope you have seen the structure and the processing of the tRNA and the chemical modifications of tRNA. See you next time. Thank you.